Okay, so I'm pretty sure we're taping. We're going to talk about ribisco, ribulose 1,5-bisphosphate carboxylase. It's the most abundant protein in the world. So this is, without this protein, we would all be dead. Can anybody tell me what they think it does? Nobody wants to talk now that we're taping. Let's talk before. Any ideas? What would it do? Without it, there'd be no cheeseburgers. There'd be no going to Subway to get a sandwich. Just something really important. What?
Carbohydrates.
It has to do with carbohydrates, right? Take something out of the air. Where do plants get their sugar? CO2, carbon dioxide. We take carbon dioxide and we make it into sugar. And all plants do this. So all plants have this protein. And the protein has a weakness. It binds the CO2 to make 3-phosphoglyceraldehyde. And this is where the term C3 comes from. This is a 3-carbon C that is the primary mechanism for photosynthesis in cool season plants. It's also the primary mechanism for photosynthesis in warm season plants, but there's another protein that's not in the cool season plants that's a 4-carbon protein that we're going to talk about. And again, this will all be up on the video, so you'll be able to see that. If it grabs CO2, it makes 3-phosphoglyceraldehyde. If it grabs O2, it forms 2-phosphoglyceraldehyde. 2-PGA is not adding anything. So it's like the protein is blind. And one of the cool things about proteins is proteins are structural. So DNA makes RNA, makes proteins. Then the proteins fold up to a shape. And the shape has a hand. You can picture if I have a hand and I'm in this room and I'm trying to grab, the girls are CO2 and the boys are O2. I'm trying to grab girls. In this room, and I'm blind, I'm going to grab a lot more boys. And I'm going to spend energy trying to do that. It costs me. But I'm going to get no energy from that. That process is called photorespiration. It's wasted energy. If I grab Matt, that's wasted energy. If I grab Ann, I can make sugar. But once I grab Ann, I can't grab her again, so it's going to be even harder to grab a woman. There's only going to be one left, so I'm going to have to grab 10 or 12 guys before I grab a woman. Warm season grasses are much more efficient than that. Does that make a little bit of sense? So C3 plants, cool season grasses, C4 plants, warm season grasses. Okay. So here's some definitions. Definitions are important. Photosynthesis, making sugar for plants by fixing CO2. And you should all have an equation for that. You should know that equation. You should know what all those chemicals stand for. So we've got our C here, and then we've got 2 double bonds to O. So that's what that molecule looks like. But it's going to grab it by this. And you can see why it can only grab it by that, because that's what's sticking out. So it can't tell the difference between an O2 or a CO2. Photorespiration is when it grabs an O2 instead of the CO2 and it wastes energy. One of my former students said it's like going on an Easter egg hunt, finding a bunch of Easter eggs, and when you open them up, there's no candy inside. I thought that was a pretty good analogy. It's like spinning your wheels in the mud. So it fixes the CO2. It does a bunch of processes that take energy and waste it, like carrying water up a hill in a sieve. That's a biblical story, isn't it? They were put in purgatory to have to carry water up the hill in a bucket with holes in it. That wasn't Jack and Jill, that was somebody else. Right? So I'm going to go on ahead, and remember, this is going to be up, and we'll know if it's going to be up or not. So I'm going to skip this and come back to it, but you want to know C3, cool season grasses. So the bluegrasses, the ryegrasses, and the tall fescue we're talking about. C4, Bermuda, and zoysia grass. See, the structure is a little bit different. I have a better picture. But let's get these basic terms down too. So C3 versus C4. Rubisco is an enzyme that catalyzes CO2 to carbon, 3-PGA. It's in all mesophyll cells. Mesophyll cells are the cells in the leaf. The bundle sheath. This is a new term we're going to talk about. The bundle sheath is only in warm season plants. Very important. The bundle sheath is a structural thing. It's a place where the warm season grasses puts all of the rubisco. It keeps its rubisco in a small place. And it uses a different protein to superload that place with CO2. The picture is going to hopefully drive that home a little bit. Pep carboxylase is that other protein. It's only in C4 plants. And pep carboxylase can grab CO2 by the C. So it knows. It's got eyes. It can go right in there and get that CO2, and then it throws it into the bundle sheath. So it superloads that area with CO2. So because of this, photorespiration does not take place in warm season grasses. Warm season grasses don't waste energy. When their stomates are closed, they still can photosynthesize at a productive rate. When cool season grasses in the summer have to close their stomates because it gets too hot, they rapidly fill up with O2, and they photorespire, and they get wet wilt, and they get sick. We don't get that in the warm season grasses. So it's kind of a combination of light and moisture and a bunch of stuff, but it really, the bottom line is the opening and closing of stomates. Cool season grasses cannot photosynthesize with their stomates closed. Warm season grasses can. So here's an example. We've got these mesophyll cells. This is rubisco is this dark color. So in the cool season plant, this is say this is a Kentucky bluegrass. The rubisco is everywhere. So it's grabbing, it's blindly looking. Here we've got our pep carboxylase surrounding the bundle sheath. The bundle sheath is filled where all the mesophyll cells are. So we force CO2 concentrations to be high in here. So the rubisco is always grabbing the CO2 that it can grab, and it's making sugar much more efficiently. So it can grow much faster when there's adequate light. When there's not adequate light, it's at a disadvantage to the cool season grasses. They can have their stomates open and they just can throw more paste at the wall. They can photosynthesize faster if those stomates are open and it's cool. If it gets hot and they have to close up the stomates, they cannot compete with the warm season grass. Does that make sense? So stomates are really important. And there's the stomate close. And this other picture has the stomates in it. And that's why I wanted to talk about this. So here's our cool season plant. It's got this mesophyll all over. The stomates are open. The CO2 has to diffuse through here. And it's being used up. So if something's being used up, there's a lower concentration of it. In the warm season grass, the air comes in, the pep carboxylase grabs the CO2 by the O and it forces it into this bundle sheath where it can photosynthesize. The bundle sheath surrounds the xylem it can quickly put, or surrounds the phloem, it can quickly put that sugar into the phloem and move it to where the plant needs it, which would be the stolons and rhizomes and the leaves, so it can grow much more efficiently when there's adequate sun. And that makes sense because the warm season grasses evolved in places like Africa, Japan, where it's warm, where the cool season grasses evolved in places like England and Scotland, where it's cool. Does that make sense? I'm going to stop the video there, and then I'll answer any questions you want. We'll make sure that this worked.